nothing, because it's not going to react with the protein. But if I put it in you, what happens even in your mouth, the sucrose?
Breaks apart.
You break it down. You start digesting that. And that means for you, you have the enzyme to break that bond, and it becomes a reducing sugar, even though it was, as the disaccharide, it wasn't. You break it into monosaccharides. Okay? So, good for plants, bad for you. All right. These were the modifications we talked about. So let's review really quick. What are we trying to do here? Let me quickly pull this over. Describe how carbohydrates are classified and be able to distinguish between different classes. Now, this is a big field. There's a lot there. So far, you've talked about aldoses and ketoses, trioses, and ones with 4, ones with 5, ones with 6. Gosh, there's different ones. We talked about furanose versus purinose, okay? Reducing versus non-reducing, alpha versus beta on the anomeric carbon, disaccharides, monosaccharides, polysaccharides, homopolysaccharides, heteropolysaccharides, and there are more that we're going to get through. So this is a big group, okay? Identify the anomeric carbon in a monosaccharide, disaccharide, polysaccharide. You should be able to do that now. Some of you were just checking into how to do that for sure now that it's moving into disaccharides, and it'll be the same in your polysaccharides, okay? Memorize the structures of the alpha D glucopyranose and the alpha D fructofuranose. I don't care whether it's the alpha or the beta, right? Because that really doesn't matter. That's what we're talking about. And ribose. 2 possible anomers that can form when a carbohydrate ring, that's the anomeric pair, right? Okay, good. What chemical modifications can be commonly seen in hexose derivatives? That's the ones we just barely looked at right before I pulled this up, right? Mm-hmm. What do I need you to learn there? Just recognize the function groups. Okay, so if you're looking at it and you're like, hey, I'm looking at this, yeah.
I read in the book somewhere that it happens a lot on carbon 2.
It does.
Is it always on carbon 2 or just?
Carbon 2 is really, really important. It's also really important, you'll find out with your Y-DNA has that hydroxyl removed, okay? But it's a very reactive site for us to play with, and it's one that's selected for highly, okay? Carbon 2. Carbon 6 and carbon 1 are also very prominent positions.
Do you also know how to name the chemically altered sugars? Or just
Yeah, great question. So again, when we look at, I'll just pull that slide up right behind us, right? When we look at this list, what do I really expect? I don't need you to say, this is glucose 6-phosphate yet. Meaning second semester, boy, you're gonna, that's like, yeah, of course, for that. But I hope that you could know that was the 6th one, and that's a phosphate. And since you should have memorized the structure for glucose, yeah, I would hope you could get that right. And that you could say whether it's beta or alpha, right? That should be doable. But I'm not focused on that aspect, but I do want you to be able to name a phosphate. But when I come over here, what are we looking at there? Could I name that, which is now named a beta D glucosamine? They could have named it on the 2, right, for the amine, but they don't mention it. Why? For the very reason you just said. It's the super common place for the amine, which also means it's the super common place for what?
The
acetyl group. Yeah, it is. And so as we look at these, you start looking at, oh yeah, it's a super common location that we start looking at things, okay? This location right here, also super common, right? We're talking about the 6th position, okay? The lactone, don't need to do all that. So I don't need all of this, but I do expect that when we are talking about it in class, and I'm talking about something where I'm saying it's an acetylmuronic acid, well, what's the acid? It's right here, right? Mm-hmm. It's like, oh, it's a carboxylate group that's been linked with an ester. Okay, I get it. Meaning you should be able to handle functional groups to see that's acidic, to see that's got an acetyl group. That's what I'm interested in. That you know what I'm communicating when in just a minute, we'll start using those modifications to decorate our sugars. So it's not so much about naming it as recognizing the functional group that you could match it to if you were in a multiple choice. It's not on the write-on. You know it's not on the write-on if you've reviewed the write-ons. So it means it's all about, could I recognize it enough to handle the name? And I don't need to make it very confusing. All I'm after is, can you recognize the functional group for the most part, okay? All right. We talked about this one being important for a paper. We'll come back to it, right? Let's check over here one more time at our list. Reducing, non-reducing, check. Why can we distinguish it? Why is it important to? If we just talked about non-enzymatic glycation, yes, we will be talking about enzymatic glycation. And we're going there, okay? Types of glycosidic bonds between carbohydrates, identify them. And then we're talking about all these polysaccharides, which is where we're going today. Okay? So let's see what we can learn. This list right here is the summary slide of the next few we're about to learn. We'll talk about them for just a brief moment so you know what we're talking about, because these are common polysaccharides that are often talked about in biological systems, and I want you to feel like you know something about it, okay? So glycogen should no longer just be this, it's a storage molecule full of sugar. But you're gonna be thinking about, oh, I know what the linkages are, I know what the branching is, I understand that that is more dense than it would be for a amylopectin. And knowing the similarities that must be between that and amylo. Okay. All right, so that list in green, definitely memorize those linkages, which makes these ones easier. So you look at this one, and I just said this was cellulose. If you were paying attention, you would already have memorized the value, but let's see if we can name it anyway. What is that linkage?
So
Tell it to somebody else. Tell it to somebody else. What is that linkage?
It's 1,4. And it's beta and beta.
Yeah, now.
Isn't it nasty? They just turn things over on you, right?
Mm-hmm.
They do this.
It's
Only
beta
think
and
about the
beta.
linkage, don't
Yeah,
think
'cause
about
it's
everything
cis,
else.
right? So you have the flag that's axial on the left.
Can
And
you
then
identify the
bond is
anomeric carbon?
axial for that carbon.
And is
And
that
it's equatorial.
cis or
for the other
trans?
one, and equatorial.
Okay. If you said beta 1,4, you are right. That is cellulose. This linkage right here, you cannot break. You don't have enzymes to do it. Okay, cool. So it turns out that the ruminants, which means what?
Cows.
Cows, goats, they can't break it down either. Termites, they can't break it down either. But bacteria that reside as symbiotes in termites, bacteria that reside in ruminants, they can.
Good for them.
But you can't.
Heroes.
Okay? So termites can break it down, cows can break it down, beta 1,4, symbiotes in that, and you, that's a no-go.
Put a big ol' X.
Yeah, okay. Cellulose is rather interesting. I should have brought my piece in. This is a really cool question, and it's a really important question. Cellulose is sugar. It's glucose. Many glucose molecules are soluble in water. They dissolve in water. Why does cellulose not dissolve in water? Talk about it in your group for just a minute. Why does cellulose not dissolve in water?
Well, that last slide showed a lot of H bonds.
All the answers you need are right here, but yeah, talk about it. Like, I don't know if I can get it out of that, but they're cool.
I mean,
Isn't it just like the way it's bonded to
I
each
think
other?
it might also have to
Yeah,
do with
so
if
maybe
you have really
make
long,
it so
if you
like
have a, if you have a, I think I remember reading
everything that
if you have
would
a
be
beta,
cool.
it's followed by a beta.
So
Linkage,
how
it's
the
like
lines
creating
of
a zigzag or a straight line.
itself,
And
then
so you
it
can have
won't,
like
like
stacks of
as
these
a
straight
whole
lines on
thing,
top of each
it's
other.
non-polar.
So not only are the individual molecules, but
I just made that up.
clumped
Yeah, just in the
together.
next slide, it's that hydrogen.
Well, what's it say?
Like they'll hydrogen bond
Hydrogen
each
bond
other over
formed between
water.
NJC monomers and additional hydrogen bonds between linear chains. So they can't really grab on with water. Yeah, it says water insoluble.
Talk about it for just a minute, you get something? Okay, I know one group got it. Anybody else want to volunteer they got it? Not sure? Go for it, what do you get?
It's the hydrogen bonding between the glucose itself that's preferential to the hydrogen bonding that water will try to attack.
That was so well said, you guys, that was like a home run. It prefers to hydrogen bond to itself in the way the structure was created than to hydrogen bond to water. It's already happy. And it's locked in place. That's super valuable. Why?
You should have cell walls that don't dissolve, I guess.
Cell walls that don't dissolve. That's like a great idea. And that means we get to build all sorts of things with it from there too. Okay, so that is the secret. Turns out that in order to do that, as it excretes it, it doesn't send out just one long beta 1,4 linkage. What it sends out is a rosette. In fact, it doesn't send out the rosettes, which are actually 6 of them at one time. It actually sends out an entire 6 times 6 of those together that it guides. And we study that in the second semester and how that's created for plants. But that's how it's creating its cell walls around the plants. And it's done outside of the cell membrane. And it controls it and creates it. So that's really kind of cool. So those are called little rosettes, and that's how we do that, beta 1,4, and then it hydrogen bonds to itself versus water, and that makes it really cool. Now, if you were Brother Johnson, why would this have been important? Does anyone know what Brother Johnson did for his career? Yeah.
He helped design glue that would hold together like plywood, basically, if I remember right.
That's right. And his whole industry, in fact, when you go to Georgia Pacific and you see in the bathroom, what do they make? Paper products.
Yeah.
Wood products, adhesives for that. It's all about this question of how do I get things to work? And so did he have to understand the chemistry going on here?
Yeah.
Yeah, so they break it down so they can actually solubilize it, so then they can relayer it into plywood, then adhere the plywood to each other. That's all part of the game. So there's a lot of things in cellulose there to learn. Cotton industry, you gotta understand it? Yeah. I think this gives a whole new world to them. Awesome. Glycogen.
What is this?
Glycogen is the storage molecule that we like to put. In the center of glycogen is a structure. It's got squigglies. What are these squigglies?
It's like proteins.
That's protein, that's an alpha helix. All right, we're gonna guess right-handed, right?
Sure.
Yeah, sure. It's actually glycogenin, which is at the core of every glycogen, and it's able to synthesize 6 on its own self where it attaches it. What linkage? What is the linkage that it's linking it?
Alpha.
Let's look right here.
That one's,
Okay, so glucose,
that's alpha.
that's alpha. This is another glucose, that's
Alpha.
alpha 1,4.
Yeah.
Oh wait, beta 1,4, alpha 1,4. My, it's so easy. It's all about the 1,4s. This is great. I'm hanging out there. Okay, so that is glycogen. It also has alpha 1,6. So the glycogenin only adds 6 on it, and then it's done. It can't reach around to keep adding more, so it comes in with other enzymes. There's a set of just another 3, and they will keep extending. But in addition, once the chain gets going, every 8 to 12, it adds a linkage at, you've named it, an alpha 1/6 position, and those branches occur, and those branches allow it to create this structure. Now, one of the researchers in my institution at Montana State, she's an organic chemist, and her whole career was about building dendrimers, molecules out of sugar, to make them sized just right so that she could hit receptors and hit not one receptor but 2 or 3 with the size, and so she could test how well the receptor was, and she can use them as transporters for hitting certain drugs and certain targets. Whole career based around just making carbohydrate structures, and how many generations wide is it, so you could control the size, and then how can I decorate it. So what is the value of this? Is this a reducing sugar or non-reducing sugar?
So it's reducing sugar.
You always have the open ends on the chains.
It starts by linking, but on the end, what's there? Is there anything on the branch chain there?
Wait, are you?
On the branch, if this hadn't added, and that was just a hydroxyl, would this?
No.
Non-reducing.
It's non-reducing. You're storing your sugar in a non-reducing form. Where's the reducing end? Ultimately, all attached to either the other sugar or
Glycogen.
to glycogen. That's where we stuck it all in. So, in order to get it off, I have enzymes that chew it off. But why is it beneficial that I made it branched?
So you can
This is
store
a
more.
kinetic question. Why do it that way? I can store a lot more for space. That's valuable. What else? So, you got it? Okay, go for it.
It makes,
Natalie.
yeah, no.
Because making bonds, reduces allergies.
Okay, so if I am gonna release all that, I'm gonna release the sugars. Go ahead.
There would be more places for the enzymes to start breaking it down.
That's the secret. I've exposed more ends that the enzymes can chew on. Instead of doing it one sugar at a time in a linear one that was super long, I've got literally hundreds of ends I can chew on, which means I can break down the sugar very, very fast and release a whole bunch of single monomers that then I can do something with. Cool, okay? So glycogen itself, great way to store things. It turns out it's primarily, we talked about, stored in the liver, which is 7 to 8% by mass. It's also stored in the muscle cells, 1 to 2% by mass. Here's the kicker. This is the one that throws us in that. Now, we talked about why not store glucose in its monomeric form. Let me give you some real values. Osmolarity. The glucose equivalent of glycogen stores would make glucose concentration 0.4 molar. For reference, the typical osmolarity of a cytosol is about 0.01 micromolar. What would happen?
Chaos. Would destroy your cells.
Talk about osmotic difference, right? That would be a massive mistake. Okay? Okay, so big deal in osmolarity. How many molecules of glucose would we predict existing glycogen stores if the blood volume is 5 liters? Well, we just talked about that number. Okay, 0.4 molar. That means 0.4 moles per liter, and I've got 5 liters of it. That would tell me how many moles I have, right? And then I could kick that out, and I could figure out how many, avocados number. How cool is that? I used to do that in 105. Okay. This tells me how many glucose molecules, which if I translate that into grams is about 360 grams or 0.8 pounds of glucose.
That's just in your blood though, right?
That's actually the equivalent of all your glycogen stores.
Oh, into your blood. Yeah, okay.
Meaning we don't have, that's what's stored in your body.
Got it. Cool.
Now, interestingly enough, this is also informational. An adult weighing 70 kilograms stores roughly 400 grams. There's the stated value from a wiki, okay? Of glycogen in the muscle, 1 to 2%. And an additional 100 grams in the liver at 5 to 6%. So where is most of your glycogen stored? Is it in the liver or is it in the muscle?
Muscle.
But they're not very concentrated in the muscle,
There's just
but there's a
more.
lot of muscle.
Yeah.
Why would I want it in the muscle, by the way? Easy access to the energy that I'm about to use because I'm working out. That's good. And the liver is the next location we'll break it down. Okay, starch. This is plants' place to store it, right? They come in two forms, amylose and amylopectin. When you think of pectin for making jam, it's the same thing, okay? Amylose is alpha 1,4, amylopectin is alpha 1,4, but its branches are at alpha 1,6. But instead of it being like it was for glycogen every 8 to 12, it's now every 24 to 30. So virtually the same thing, just in a lot less dense. Some of the amylopectin up to 200 million in molar mass. Big. There's these guys we just looked at there. Linear form versus branch form. Cool. Here's some gee whizzes of these, but also interesting to recognize. Huh. Are there three-dimensional structures when I have long strands of amylose or amylopectin? Yeah, actually there are. And it turns out that this part right here in the cyclase form is moving and rotating around this glycosidic linkage. What do you notice by the glycosidic linkages?
It's similar to proteins.
Similar to proteins, it's got phi and psi angles. And if I keep rotating around in a circle like an alpha helix, that's typically how we store starches. And it's interesting, but iodine intercalates into that just perfectly. So that when I was a boy and iodine was easily available for anybody, instead of meth world that we now live in, which is why it's harder to get to, we would take potatoes and we would dip them into iodine, and what would happen to the potato? It would turn what color? So you know iodine is a solution brown if you've been to the doctor's office, but what happens when it interacts with starch is it turns purple, which is why in the lab when I do a test for starch, which is an iodine titration, I'm looking for the purple. Huh. And this was the way we would paint even on our papers in kindergarten and first grade, you guys.
Oh gosh.
And what else would turn? If potatoes did it, what else would turn? Any starch. It's an indicator for starch.
Really?
Cool. Isn't that fun? You're like, iodine, I'm getting some of that. And it can make meth, too. Cool.
Oh, now I know that.
Do these have three-dimensional structures?
Yeah.
Yes, they really do, okay? Starch, glycogen, cellulose. These have structures and they have form. That form is important to us, okay? Here's some gee whiz facts that will not be on the exam, but I think are interesting because, well, frankly, you guys should be chemists. If I'm looking at amylose itself, it's generally insoluble in cold water. Just straight chain starch. It's more resistant to digestion. Why? Because when it actually does condense, it actually wraps around itself in helical structures and makes packed molecules that are hard to unravel. So what do you do with starch in hot water?
You unravel them?
You unravel it, gals, what do we do with starch in hot water?
So you can make sauces if that's what you're talking about?
I am talking about that. I could use cornstarch, and what can I do with it?
Make it
Thinking.
I could make gravy. Okay, that's cool. Is that the only thing we can do with it? No, I can make a whole bunch of other things that it's a thickener. When does it actually work?
once you've heated it.
When I heat it up, I unravel it.
Oh, wow.
And that's, oh yeah, my potatoes cook. And that allows it to absorb water where it was actually hydrogen bonded to itself. Now I'm unraveling it. So it's not winning the cellulose game, just a little bit of heat breaks it apart. Amylopectin is in both hot and cold water, is not resistant to digestion, okay? This one turned blue-black with iodine. This one's a reddish-brown. Glycogen itself gives a red color with iodine. It's soluble in water, easily digested. Cellulose, not soluble, doesn't give any color with iodine, right? Tensile strength, almost equal that of steel in the fibers. So there's your starch discussion. Different starches based on the different length and the fact that I get pectins that also combine with also my starches. This is what you're looking at, these wrapped up strands then heating up. So why does my bread get stale? If I heated it up and it unraveled and it picked up water, what happens to make it stale?
Is the water evaporating?
The water's leaving and then it's getting wrapped back around itself and that makes it hard again. Oh, what happens when I throw my gravy that I made with that cornstarch into the fridge? Some of you would never store gravy, but what does happen?
It gets hard again.
What's on top of it? Water. Where did it come from? That was what was in there. Now I've cooled it down. It's recrystallized. My gravy's all lumpy and I have to heat it up again to get it to work. That's what's going on. That's stale bread, which means what's going to happen to your bread when you throw it in the fridge? This is a great lesson for my wife. She's like, she makes me homemade bread all the time. Guys, you should marry a wife that makes bread all the time. This is a great life, right? Okay.
If you put it in the fridge,
it will go stale
faster.
Very quickly. How quickly? As soon as it gets cold.
As soon as it gets on
It's stale. Why? Because it's forcing the water out. That's what's happening. Okay.
So it'll last so much longer.
Yeah, and different ones obviously at different lengths, so different temperatures to get them to work, whether it's tapioca or potatoes or cornstarch or wheat flour. Okay. There's all that breaking up, unraveling those. Okay, you can see how they combine together. Nobody wants to go to dental school in here, right? This is for you guys. Bacteria. You guys know these as what? Strep, right? Did you know they live in your mouth?
I mean, yeah.
Did you know that that plaque on your teeth is made by them?
Really?
As they do, they make biofilms. They do quorum sensing. They're living and they're layering themselves. And to adhere to your teeth, they excrete a substance that they form from sugar. And that substance actually that they excrete is dextran. And it's a polymer, and it turns out that that linkage is right here. Okay, so it's an alpha 1,6 with an alpha 1,3 branch, and it turns out that you don't have the ability to break that down. No enzyme for it in your mouth. So what's that going to do? The bacteria can break it down for food, but you can't. But it allows them to sticky on your teeth and you get all that tartar. And then of course what happens? Acid buildup in cavities, and yep, that's where they hang out and live. So what would happen if I gave you their enzyme and put it in your mouth?
You get diabetes. Because you'd have too much glucose.
It's hardly any sugar. But yeah, you could leave that way. No. What could I do?
Break it down.
I could break that down. Talk about going to the dentist, and a hygienist doesn't have to do any scraping. I'm just going to swish this for how long? Well, if I did it every day, I'd probably be awesome. But if you don't do it every day, hours. Nasty. And what's your pH and what's your salinity and is it right and is the enzyme going to keep working? And yeah, but they're experimenting with it and they continue to experiment with it and they have different ones that work at different rates. Okay. You're like, I'm putting that in my toothpaste. This is going to be awesome.
Yeah.
Yep, I know.
That's what I would do.
chitin. I got money to put chitin in my mineral brines that we got from the Great Salt Lake because importing them into Korea required it to be a manufactured process as an import. I couldn't just ship something that wasn't manufactured, so we had to, at their council, you need to do something. Why don't you add something like chitin? What is chitin used for?
It's pretty like lobsters.
It's the external exoskeleton of lobsters or crabs or also bugs, insects, right?
Yeah.
So is it soluble in water?
No.
No, it's not soluble, okay, in water. So what's its linkage?
Alpha. Or sorry, no, that's beta. And beta.
Yeah, it's a beta
1,1.
Beta 1.
beta 1,4. Wait a second, if this is beta 1,4, beta 1,4 is the same stuff that cellulose is made out of. Is it wood? No, what's different?
It's got the
What's this
acetyl,
function group?
N-acetyl.
N-acetyl. That added to it makes it different.
Why is it 4
Okay.
and 1?
Alginic acid, Dennis, here I'm looking out for you right there. There's the stuff you want to put in there so you can make a modeling agent. Put it in the top of your mouth, make this awesome frame. Ooh, look at their teeth. This is cool. Yeah, there's what we're doing. Agarose, anybody use that?
Yeah.
Yeah, where did it come from?
Seaweed.
Seaweed. Ooh. I can grow my bacteria on it. I can do gel electrophoresis for my DNA. Cool. Agarose. Here's chitin. We've already seen the linkage on that. Okay. Cool. Similar to cellulose. Tough, more flexible. Agarose. Wouldn't that be a great job? What's your job? Go gather seaweed so I can make agarose gel. That's cool. Thinking of your new career. All right. Here's your agarose. Cool. That was it. That was your polysaccharides. Now we're going to move into glucosaminoGlycans. We're going to pick up some speed because you don't have to learn as many details here. GlucosaminoGlycans. You ready? What did I say is the distinguishing feature? Big green arrow, probably on the test. Make a note of it. One monomer is either what? It's got a N-acetyl group on it, or it's got a N-acetylgalactosamine. Okay, so that is defining in a repeating disaccharide unit that it's a glucosaminoGlycan. They typically have negative charges with sulfates or uronic acid. Okay, it's used in framework like connective tissues, lubrication for joints. So let's learn some cool things. Here's my eye. Have you ever wondered why your eye isn't dried out? Some of you are like, it does get dry and it hurts. Why isn't it dry? Why does it have moisture that appears on the surface of your eye?
I mean, isn't that what your tears are for?
There's sugar on the top of your eye.
Out of town.
Hyaluronate, I won't name the enzyme. Hyaluronate, though, is found in lubricant for your teeth. It's also on coating for unfertilization. It's also on your eye. And it's a sugar. And because this sugar has this group right here, and this group right here, what does it allow it to do? Pull in waters. So as we coat your eye with that, it starts pulling on waters. Helps it stay soluble. Not soluble, but wet and moist on your surface. So yes, your tears are doing it, but why aren't they drying now? That's why. So this is actually the same hyaluronate that's put on an egg. So when you learn your biology lesson, this is a cool side note. Some people think I'm weird because I know this stuff, but we learned that there was some coating on the egg that the sperm had to somehow get through. It was the hyaluronate. And on the head of the sperm is what? An enzyme known as hyaluronase. And it chews right through it.
That's pretty good.
All right. So now you're learning some cool stuff, right? figuring all that out. This one right here, I sold in industry, okay? I didn't believe in it, still don't believe in it.
You sold it?
My company did, right? My job is to test it.
Like, gotta eat.
This is called glucosamine sulfate. So again, what are we looking at? Repeating disaccharide units. This one's 20 to 60. I could have pointed out the other one, you don't need to know these, all these, we're just pointing them out, and you're able to identify what are the key characteristics. This one has uronic acid, it also has sulfates. Again, remind yourself, this is how I categorized it. This was the one in green. What is it that we're now showing you? So these are examples of, which you don't need all the examples, you need the definition for what it is, okay? Gonna have either an N-acetylglucosamine, an N-acetylgalactosamine, gonna have some uronic acid, which I just showed you is just a carboxylate group, or some sulfate esters on it, okay? Wow, okay, there you go. Was it there? Yep, it was there, okay? So, beta-1,3 linkages, put in here. What's awesome about this stuff is this is what cartilage of shark is made out of, and what do we use it for? It's supposed to be good for your joints. Let me tell you my criticism of it, and then you can treat it as a biochemist logically. How is this gonna get through my stomach and inside of my bloodstream if I ingest that? How long is it? 20 to 60 in length. What am I gonna have to do to cross a membrane?
Be uncharged.
Be uncharged. What else am I gonna have to do?
It's non-polar.
I'm gonna break it down to single monomers. Yep, I'm gonna have to be non-polar. So all that has to happen to get it ingested into my body. So is this going across the membrane?
No.
The building blocks might be, but not this. And to some degree, very different for it just to get absorbed. So what am I eating if I spend money on my glucosamine sulfate? Sugar.
Well, it's a sugar.
Expensive sugar that I killed sharks and ground them up for. Awesome.
Literally a sugar.
Let's just do it. Yeah, sharks are bad.
Just send it.
That's
Okay.
funny.
So there you go,
Holy
absorption,
cow.
okay.
Get wrecked, shark. Poor guy.
I'm challenging you to actually ask the questions instead of just believe stuff. Start asking. So I'll try to teach you truth and what's not. By the way, some people swear by this.
Gives them energy.
It seems to make their joints feel better. They're just like, "Oh, my knee hurt forever until I started taking this." What are they experiencing?
Sounds like
So
snake oil.
smart. Yeah. Okay. So here's another one. Found in your cornea, cartilage, and bone. Keratin sulfate. Okay. Sweet. There's another one. We're going to point out this one because it's important in medicine today. It's also important in biology. This is known as heparin. Okay, it's also a glycosaminoglycan, okay? Anti-clotting agent. See, what do you notice all over this? A whole bunch of sulfates and uronic acid or carboxylate groups, right? It's the most highly negatively charged biomolecule we know of.
Wow.
It is 15 to 90 of length, this heparin. It's known as an anti-clotting agent. Did you guys know that? So we actually, when you get your blood taken, sometimes we pull it in and it goes into a heparin that we put inside of the tube so it doesn't clot. Huh, that's pretty smart. Yes. There's its length. Yes, highest negative charge. It's in green, you should maybe remember that. Maybe you should really remember that, okay. Regulates the development and formation of blood vessels. Also might affect virus and bacterial, decreasing their virulence. How is that possible? Here's heparin itself, just pointed out. We are going to look at it again in just a few minutes as we move forward. And what we're going to see is that heparin actually, because of its charges, has specific binding sites. And it's really nice as a sugar-coated molecule that we have it all over our body, so that we can release it when we need to, also so that we can use it to signal that's where this is, that's where it is, because it does some awesome conformational changes, okay? Viral proteins are heavily glycosylated, helps evade the immune system. Cool. Half of all mammalian proteins are glycoproteins. Wow. Okay, so these are now talking about glycoproteins. So this is different than the glycosaminoglycans. Now I'm talking about a protein with a sugar. Okay, cool. So we'll call the class that you should know the group, even though we're going to look at examples, a proteoglycan. Okay. Glycosaminoglycans are linked to the core of the protein. We just looked at glycosaminoglycans. They're linked to the core of the protein, okay? Usually from a xyloserine hydroxyl, so serines. What's the functional group?
SNOH. Hydroxyl.
And then we're going to use this fancy word, aggrecan, as the core of the protein. So, non-enzymatic glycation, this is enzymatic glycation. You see a star that's in green?
Mm-hmm.
That means it is on the exam. Oh, wow. I made it as transparent as I can. If you see more stars, they are on the exam. Okay. So this is O-linked, this is N-linked. What are the amino acids if I'm O-linked?
Serine.
Serine. So this is the sugar connecting to a serine. And an enzyme connected them. How would it have to connect? What has the serine got that will allow it to connect with a sugar?
Hydroxyl.
The hydroxyl group's lone pair is attacking where?
The carbonyl.
The open form, anomeric carbon, is where it can enzymatically be attached. Oh, that makes perfect sense. Great. How about the N-link? Asparagine? Beautiful. Threonine, you notice, was also here.
Yeah.
So threonine, serine, and here asparagine. Only asparagine is being listed, though. Even though there are other amines that you could think about. The enzyme selects for asparagine. And it will link them through the nitrogen's lone pair with the anomeric carbon. So that's pretty cool. Should you know those specific amino acids? Yeah, because those are the only examples of what are called O-linked or N-linked. And enzymes do that. When I put them on proteins, I could call it a glycoprotein or a proteoglycan. Which one's which? This is like meant for the super intelligent to make you feel like this is really harsh, but if I have more protein, it's known as a glycoprotein, and that must mean by mass there's less sugar. So I'm modifying my protein, which I have most of, with a sugar. If I have more sugar, and then I'm attaching a protein to it, we'll call it a proteoglycan. Ah, made my life easy. Okay, there's our aggrecan core. That's pretty cool. What do I see coming off of the aggrecan core that we just talked about? Hyaluron, that's right. This is the same stuff that picked up all that water. Ooh, it's all fuzzy. These are all those sugars.
So those are all places where you can attach a bunch of waters.
Like I can attach a lot of waters, and I can put this in my cartilage, and it's going to hold all of that. It's like a caterpillar,
wow.
but it really is not. Okay. Chondroitin sulfate thrown into two. Nice. Okay. By the way, gee whiz fat. If I talked about a glycolipid or a lipopolysaccharide, same thing as we just talked about with the proteins. So I can modify lipids too. We have to talk about those. And then we will talk about the sugar code come
Monday.
Monday.